Good afternoon, students. Today we are going to study the new topic, that is ultrastructure of bacterial cell. Bacteria are unicellular organisms that have a variety of sizes, shape, and envelope structures. The minimal requirement are cytoplasm, a cell membrane that surrounds the cytoplasm, and a DNA chromosome. A few have internal structures such as vacuoles and storage bodies, but none have true organelles. In this presentation, in this slide, you can see the diagram of ultrastructure of bacterial cell, in which a bacteria cell is focused, which shows the ultrastructure. Whatever organelles that are present, they are described here. You can see that it is a diagram of bacteria cell, in which it has various layers. Outermost layer is called capsule. Inner to that there is a presence of cell wall. Inner to the cell wall there is a presence of plasma membrane. Again, structure is there. Folded structure is there, that is called mesosome. Inside the cytoplasm there is a presence of nucleoid, that is DNA, which is present in the suspended form. Again, plasmid is there, that is extra chromosomal material is there. Again, some inclusions are there, polyribosomes are there. Again, at the outermost layer, motile structure is present, that is called flagellum. And one of structure is called pilus, pilus structure is present over there. So these are the some important we can say inclusions are there in the ultrastructure of bacteria cell. So we will see one by one in detail. So the bacterial cell reveals three layers, that is called capsule. It is also called as a glycocalyx. Second one is cell wall, and third one is cytoplasm. Okay, so capsule, in some bacteria they are surrounded by a gelatinous substance which is composed of polysaccharides or polypeptide or both. A thick layer of glycocalyx bound tightly to the cell wall is called capsule. It protects cell from desiccation and antibiotics. The sticky nature helps them to attach to substrates like plant root surfaces, human teeth, and tissues. It helps to retain the nutrients in bacterial cell. So it is very helpful structure which is present in the bacteria, that is called capsule. Inner to the capsule there is a presence of cell wall. The bacterial cell wall is granular and is rigid. It provides protection and gives shape to the cell. The chemical composition of cell wall is rather complex and is made up of peptidoglycan or mucopeptide. One of the most abundant polypeptide called porine is present, and it helps in the diffusion of solids. So it is very important to note that cell wall is made of peptidoglycan and polypeptides. Again, porine is the important what we called polypeptide, which is present in the cell wall of bacteria cell. Next is the plasma membrane. Inner to the cell wall there is a presence of plasma membrane. The plasma membrane is made up of lipoprotein, that is lipids and proteins. It controls the entry and exit of small molecules and ions. The enzymes involved in the oxidation of metabolites as well as the photosystems used in photosynthesis are present in the plasma membrane. Inner to the plasma membrane there is cytoplasm. Cytoplasm is thick and semi-transparent. It contains ribosomes and other cell inclusions. Cytoplasmic inclusions like glycogen, beta hydroxybutyrate granules, sulfur granules, and gas vesicles, they are present in the cytoplasm. Okay, so these are the some inclusions which are present in the cytoplasm in case of bacterial cell. Again, bacterial chromosome is there. The bacterial chromosome is a single circular DNA molecule, tightly coiled, and is not enclosed in a membrane as in eukaryotes. This genetic material is called nucleoid or genophore. It is amazing to note that the DNA of E. coli, which measures about 1 millimeter long when uncoiled, contains all the genetic information of the organism. The DNA is not bound to the histone proteins. The single chromosome or the DNA molecule is circular, and at one point it is attached to the plasma membrane, and it is believed that this attachment may help in the separation of two chromosomes after DNA replication. So here genetic material is just present in the cytoplasm in suspended form, which is not bounded by any nuclear membrane or any envelope like structures as in eukaryotes. So that is the main difference between eukaryotic cell and prokaryotic cell. Okay, next is the plasmid. Plasmids are extra chromosomal, double stranded, circular, self replicating autonomous elements. So they contain genes for fertility, antibiotic resistant and heavy metals. It also help in the production of bacteriocins and toxins which are not found in bacterial chromosomes. The size of plasmid varies from 1 to 500 kb. Usually, plasmids contribute to about 0.5 to 5% of the total DNA of bacteria. The number of plasmids per cell varies. Plasmids are classified into different types based on the function. Some of them are fertility factor, resistant factor, plasmids, colicin production, root inducing plasmids, and tumor inducing plasmids. Okay, so plasmids, they are extra chromosomal material. So they are present other than the DNA of bacteria or original chromosome. Which are present other than that, that is called plasmid, which is also called as extra chromosomal material, which is responsible for the different whatever characteristics of bacteria cell, that is antibiotic resistant or tumor inducing or root inducing, colicin production, so fertility factor, that are present in that plasmid. That is nothing but chromosomal material, but it is called as extra chromosomal material. Then mesosomes, these are localized enfoldings of plasma membrane produced into the cell in the form of vesicles, tubules and lamellae. They are clumped and folded together to maximize their surface area and helps in respiration and in binary fission. And next to the mesosome, there is presence of polysomes or polyribosomes. Poly means many ribosomes. The ribosomes are the site of protein synthesis, and the number of ribosomes per cell varies from 10,000 to 15,000. The ribosomes are 70S type and consist of two subunits, that is 50S and 30S. The ribosomes are held together by mRNA and form polyribosomes or polysomes. Okay, so 70S type of ribosomes are present in case of bacteria cell, and again protein ribosomes, they are the site of protein synthesis. Okay, again flagella is the outermost structure. Certain motile bacteria have numerous thin hair-like processes of variable length emerge from the cell wall called flagella. It is 20 to 30 micrometer in diameter and 15 micrometer in length. The flagella of eukaryotic cells contain 9 plus 2 microtubules, but each flagellum in bacteria is made up of single fibril. Okay, flagella are used for locomotion. Based on the number and position of flagella, there are different types of bacteria. So flagella is the motile structure which is present in the bacteria, and always it is always what we can say stable, that 9 plus 2 microtubules arrangement is there for each flagella. Okay, and each flagella is made up of single fibril. Okay, so and the last structure is fimbriae or pili. Pili or fimbriae are hair-like appendage found on the surface of cell wall of gram-negative bacteria. So that is important. That is you have to note that such a type of pili structure is present in gram-negative bacteria only. The pili are 0.2 to 20 micrometer long with a diameter of about 0.0 to 5 micrometer. In addition to normal pili, there are special type of pili which help in conjugation called sex pili are also found. While during sexual reproduction, these pili, they are important in case of bacterial cell. In this slide you can see the different shapes of bacteria. Coccus, diplococcus. When circular bacteria is there, that is called coccus. When two bacterial cells are there, that is called diplococcus. When bunch of bacterial cells are there, cocci bacteria, that is called staphylococcus. 4 bacteria cells are there, cocci bacteria, that is called tetracoccus. Okay, so if a single chain is there, that is called streptococcus. Okay, again on the right side you can see the monotrichous, lophotrichous, amphitrichous bacteria cells are there, number of flagella. If number of only one flagella is present, that is called monotrichous. If more than 1, 2, that is called lophotrichous. If more than these flagella hair-like structures, they are present on both the side, that is called amphitrichous. If they are present on both the on the peripheral side of the bacterial cell, that is called peritrichous, and if without flagella, then it is called as atrichous. Okay, again spiral shape bacterial cell, it is called spirillum, and if comma shape bacterial cell is there, that is called vibrio. Okay, so this is all about the ultrastructure of bacterial cell, their sizes and shapes. Okay, so thank you.